about this. All the criticism that you see on the internet, particularly Wikipedia, is by people who have never seen it at all. Never seen it, never studied it, never done anything. They write from the position of omnipotence. I have a PhD and I know better than everybody. I don't need to see it. I don't need to see it. I know what it is. It's a deformity. Because everything I've ever learned tells me that this is supposed to be a deformity. And anything that's not a deformity is not allowed in our current paradigm, so I will defend the current paradigm to the death. And that's how they do. And so everything that you will read about the Starchild on the internet, and particularly Wikipedia, is nothing but lies. Just absolute garbage. And they know better. They know we have the evidence. All they have to do is go to the website. We have tons of information that contradicts everything that they have. And yet, you still read that garbage on the internet. And people all think, the first thing they do when they meet me and they start learning about this is they run home and they read what Wikipedia has to say about it. And they say, oh, that guy's lying. That guy's a fraud. That guy's a hoaxer. Because they believe Wikipedia. And let me tell you something about Wikipedia. You have the belief that it's written by people who know what they're talking about. Okay? If you want to read about how to shoe a horse, you're reading it from somebody who does know what he's talking about. But if you're reading about it, something to do with alternative knowledge, which is what I and many others do, what you're reading there is a bunch of absolute lies. And those are orchestrated by what are called administrators, who are generally graduate students somewhere in the world with time on their hands. And they take responsibility for various aspects that they're interested in. We've had 5 that I think I'm aware of over the last 12 or so years with the Starchild, or as long as it's been on Wikipedia. And what they do is they have a thing that allows their computer to know that somebody's made a change. And they'll go in and they'll fix it back to the way they want it. So you can try your best. I offer this challenge to anyone. Go in, make a change, a positive change about it, and within a few hours it'll be changed back to the way it was. And by me doing this over the years, it's turned out, somebody contacted me not too long ago and he said, you know, I heard you say this on a radio show, and I didn't believe it because I do a lot of work within Wikipedia, and I just wanted to see. I checked your Starchild website. They keep great records. All the changes that have ever been made, they keep. And he said, I looked at your page, and he says, I have to tell you, I was really impressed. There have been more changes, pro and con, on your Starchild page. More people trying to write good things about it, and more corrections from the other side than on the page of ex-President George W. Bush. Now, that is good company of scoundrels. I mean, that is. So I take pride in that I'm that far under their skin that they have to watch it that diligently. But they do. That is the way it is. Next slide. What they say is that still to this day, you will still read cradle-boarded hydrocephaly, and now the new one, progeria. It used to be just cradle-boarded and hydrocephaly, and that stood because they could get away with it. But I've hammered them so bad at things like this, as you'll see, that a few years ago, 3 or 4 years ago, they came up with that one, and you'll see why. I'm going to go over each one now. Next slide. Cradle boarding, you all know, I hope you know, is the practice of primitive societies where you put a baby on a board to put on the mother's back in societies where the women have to work very diligently within a few days of birth. They put the baby back there, they swaddle it, and they have to strap the head down lightly, not crunch it down, just lightly strap it down so that it won't snap around like that as she works and damage its neck. Just hold it down. But because the bone is so soft over the time that it has to stay on the cradle board, it is going to flatten out as flat as the board. And the human that was found with the Starchild 900 years ago, very common, she was cradle boarded. And her back of her head is absolutely as flat as that table, about the size of the palm of my hand. It always starts right at the top of the inion. Can't go below the inion because you can't tear neck muscles off. It has to stop right there, and it stops up at the crown, and that's how it is. And flat as that. So it has to be flat. I'm sorry. Go ahead, next slide. You're right. So with the Starchild, what we see is that it isn't flat. It's got its natural convolutions. You can see them in this shot, and you can see them especially here. And if you ran your hand over it, you'd feel the natural convolutions of the bone, which bone normally has. It was never put up against a board. I mean, never. Morons and even sub-morons, poorly performing morons, can see this and understand this. But your average PhD cannot. Cannot. Next slide. Again, cradle-boarded head positions. This is when it's standard, no cradle-boarded, when they're both looking ahead. This is the angles of their heads. But now this one, if it's cradle-boarded up against the board, kicking back, mom's working, catching some rays, no problem. Everything's fine. Starchild, Mom, please. Oh. Dead in about 3 minutes from strangling. Never happened. I mean, again, sub-level morons can see this. And yet you will go right out on the internet and you'll find them saying that this is it. Cradle-boarded hydrocephalic. Next slide. Hydrocephaly, as you know, I hope, is called water on the brain. It's not water, it's fluid, spinal fluid. Normally, your spinal fluid comes up, washes out your brain of cells that have gone bad. Again, 7 years, everything but the brain, but there are parts of it that do need to be refreshed. And then it comes out through tubes that take it back and it goes out, constantly cycling through. But if these tubes are clogged or in some way impaired, it won't go out and it just keeps blowing until you die. Till the pressure gets such that it squeezes the cerebellum out of the neck hole, and that's it. This is invariably a death sentence, especially 900 years ago. Today, I mean, this is like a third world country, because today, just anybody that's born with it's a pretty easy fix to put shunts in there and allow them to go on. There are people, you don't even know what happened to them, generally. It's so easy to fix now. But again, not in all countries. But if it isn't fixed, and this is what you get, it's going to expand at the sutures. The sutures are not going to come together because that's the weakest point. They're not connected. It's easy to see. Next slide. And same thing, blowing up a balloon, you're not going to be able to put a crease in it. And the sutures are all together. Sub-performing morons can see this. The PhDs can't. You will still see them saying this is the answer to the Starchild. Next slide. If that suture is fused, you can get a crease. If it's abnormally fused, meaning just smoothed over fused. But you can put a camera, as we did up in here, to see this from the inside, and you can see the light shining through. Absolutely perfectly normal sutures all the way around. And they know it. Look, it's not like they don't know it. It's not like they don't know it. We're on that field playing that game. That's what it is. And they're trying to win. Even though they know what they're doing is cheating, they want to win. And so they just pretend that this is true, when they know good and well it's not. Next slide. Last but not least, vein tracts. When your brain has veins on top of it, and when you're an infant, your bone is so soft it touches on those and the veins leave tracks. Everybody's like this. And the Starchild has them too. Now they're repositioned in the Starchild because it has a different kind of brain, a different shape, different everything about it, but it still had veins on its brain, and it shows clearly that it didn't have fluid on its brain, separating the brain from the skull. Morons at work. Next slide. Now, the new one is, because of just how devastating that argument is against them, one of them noticed, wait a minute. The Starchild has thin bone and progeria victims have thin bone. And the Starchild has a small lower face, and progeria victim, it's got to be a progeria victim. We win, yay, progeria victim. Starchild is a progeria victim. Well, sure enough, the Starchild bones are thin relative to humans, but progeria is that horrible disease where when you're born, you immediately start getting old. And if you make it to 15, you've lived the equal of about 100. Most of them don't make it to 15. It's a terrible, terrible disease across a lot of areas, a lot of problems. But what it is mostly is you're shriveling up and dying right out of the chute, so that your bones are always very fragile because they're so thin and so weak and so brittle. Next slide. Now the star child, sure enough, you see how much thinner it is. It's dramatic. Half or less than half. But certainly in the same area here, it's less than half. And yet, it's 2 to 3 times as hard. It's much harder. When you cut these bones, if you take a saw blade and to make this bone cut right here, a saw blade, you just go zoom, 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 zoom. It'll take you about 50 strokes to make that. Human bone's not, you can see it's not thin and it's not easy to cut through. It'll take you about 50 strokes to make that length right there. To make the same length on the star child, take you about 200. It's like cutting sheet metal by comparison. You don't even know you're making progress for a while. Really amazing, that tooth enamel thing, and something else that's coming up later. But you can see very clearly, this is not fragile bone. This is not progeria victim. Next slide. Now, sure enough, lower face, reduced lower face, and you'll see this in a minute too. But it's still human. Look at where the ears are, right at the eyes where it's supposed to be. Look at where the ears are here, not even in the ballpark. Still human eyes, brow ridges, dip in the nose, human eyeballs in there. So it's shriveled up, yes, but it's still a human being. The parts that aren't really badly affected are still human parts. Next slide. And you see if in profile, brow ridge, nose dip, no nose dip, and lack of chin, very reduced because it just never gets to grow. Little thin neck, but still has the inion back here and all this. It's still just not the same. It's a human. It's a badly deformed, badly impacted human, but human nonetheless. Star child is not a progeria victim. Next slide. And last but not least, progeria victims almost never have the fontanelle close, and of course the star child did. Again, sub-level morons, anybody can see this. Anybody that's a true scientist would be ashamed to try to say that the star child is a victim of cradle boarding, hydrocephaly, or progeria. And yet you go out there and that's the criticism that you will read. Next slide. So how do they explain it away? None of the above is true. None of the above. Next slide. And if it's not a combination of what are these things, what was it? Next slide. Well, the first image we look at is by an Alaskan artist named Rob Roy Menzies, who said, look, if I take your x-ray and I put a face on it, it looks pretty cool, don't you think? And I said, well, yeah, pretty cool, except you make it look like somebody killed its dog. Can you fix that? Make it look a little more appealing. And so in the end, next slide, we got out this, which I think is really a pretty good depiction of how it probably looked. And understand with the eyes, this. With the star child's eyes, with that very, very shallow depth of its eye sockets, it can't have a ball like us. That would pop off the head like frog eyes. You can't make it through childhood with eyes like that. You have to have a greatly flattened in eye that sits there and really probably can't move. There's no way. Like we can get 180 degrees of vision by looking left, looking right, but our eyes move because they're round balls in a big socket that holds them. This, in order to get 180 degrees of vision, is going to have to be all pupil, wall-to-wall pupil, so that it takes in more. But in taking in more, you got to have a bigger brain back there to deal with all that it's taking in more. A lot of our brain works to take care of our vision, right? Star Child has all that. So I go along with this idea. Now, the standard gray idea of eyes here is very likely, I think, some kind of a lens that they have to wear over when they're on Earth to deal with Earth light or Earth dark, maybe so they can see it in the night, because you generally always are dealing with them in the night. Not that many encounters in the day. Some, but vastly more at night. So it's possible that those are lenses that allow them to see at night or whatever, but I think it's a lens sitting over the eye. Now, this communion picture, this has become the iconic image of gray aliens because of the Whitley Strieber book, the cover of the Whitley Strieber book. But in fact, and Bud Hopkins and others who deal with this will tell you, this is not what people say when they've really seen them. Bud says this is much more like what they describe. They don't describe this. They describe something much more like that. Bud Hopkins himself told me that before he died. It's pretty interesting. Next slide. Now, this is a forensic sculptor. People would say, well, has anybody ever tried to make one of those clay models of the Star-Child? And so we finally got a forensic sculptor, Bill Munns, to do it. And this is what he sent me. And I said, Bill, geez, man, those eyes, those are human eyes. It can't be the Star-Child's eyes. He says, yeah, I know. Couldn't make it work, Lloyd. They just stuck off the head and it was just terrible. I said, well, what'd you do? He said, we don't have a format for that, any kind of a template for it. I said, well, what'd you do? He says, well, I had to cut the eyeball right in half. You know, the eyeballs that they put in there, I had to cut it in half to make it fit. And he said, and then it was okay. And I said, oh, well, all right. And I said, what about the neck? What are all these big things out to the side? And he says, well, if you do the neck the real size of it, he says, you got this big honking lump of clay up on this skinny little neck. He said, if I bumped it against the table or something, the head might just fall off the neck. So I had to brace it up a little bit. And I said, oh, okay, all right. Well, that made sense too. But I mean, other than that, though, it's a pretty reasonable, pretty good look, I think. Next slide. And you'll see how he put it together, and you'll see those half eyes in there, and you'll watch the process of him putting it together, which is really pretty cool. Next slide. And we'll do it again because it's so much fun.
I've watched a forensic sculpture. There's only one thing. They put little wood pins and
right.
they build it up to a certain depth in different layers.
Some do it that way, and others...
Putting the little wood pins.
Well, nobody knows what the wood things, what they would be. But I asked him about that, and he says, no, the oldest of us do it this way. The youngest of us have learned in college to do it the new way. But he said it's the same effect. I mean, it looks good to me, other than those things. Okay, next slide. Now, this is from a guy named Chris Murphy, who, I don't know how many of you are interested in hominoids like Bigfoot and the Abominable Snowman and all that, but he does the best books and some of the best literature, but particularly the best books about that subject. And he does analysis of hominoid faces relative to human faces, and he did this with the Star-Child. It turns out that our faces are very well proportioned. From here to our chin, our eyes are in the middle. From our eyes to our chin, our nose is in the middle. From our nose to our chin, our mouth is in the middle. It just divides down like that. It's really pretty cool. Star-Child, nothing like that. Very different in each case. Now, people have said, well, could the Star-Child, something like the Star-Child, if it made an effort, could it disguise itself and walk around among us? Can there be aliens walking among us, or at least Star-Child people? And so I posed this challenge to my assistant Vivian, and here's what she came up with. Next slide. And boy, it's like, well, maybe, golly. Could happen. Anyway, next slide. Two things found in the Star-Child bone are not found in bones of any other creature on Earth. Unknown fibers, unknown red residue. Understand what I'm saying here. No other creature on Earth. You're going to see something that's not found in anything. Two things that's found in the Star-Child. Next slide. This is the cut of a Dremel blade. A Dremel blade is a blade, a whirling round blade, and a deal that you cut the bone with, and you go, whoop, you cut the bone. And if I were to have one here, it could cut my hand off my wrist in just a matter of a few seconds. It would go through. Do not screw around with a Dremel blade. Tough. So when they cut some of the first pieces of bone out, and not the first pieces, but some of the first pieces, but they looked under with a little chip with a scanning electron microscope, the person said, what is all this? What is all this? What is all this? Well, what is it? We don't know. So you never see this. This is incredible. How could it resist the Dremel blade? Well, it turns out that these are fibers. Next slide. These fibers are woven into the matrix of the bone. Here you see one of them where the blade passed just over where it lays in that track right there, like rebar in concrete, to add strength to the bone that's already super hard. Because these fibers are really, really durable. You can see that one's torn, but somehow this one came along here, it got ripped out of its track and came out here and got wrapped around this fatter one here. They're different kinds. They're different kinds of fibers. And we've had it checked from with mycologists. It's not anything that grew on it or grew there. It's not fungi, bacteria, mold, anything like that. It's in the bone. It's in the matrix of the bone. It's woven in. Not known in any other animal species on Earth, as far as we know. Next slide. We see another one here, the flat fiber snagged down here, and another kind, but what you see here up close is it's sheared off. The Dremel blade it resisted the Dremel blade to the degree that it tore it off instead of cut it through like it would do my arm. Tough stuff, whatever this is. Next slide. And here we see one absolutely clearly buried in the matrix right up at the surface of the inner part of the bone. What is it? Don't know. Next slide. Now, bone chips cut but not polished. When you cut, the bone turns into powder. When the blade is cutting, powders it, and so it blows into the cancellous holes. Human and Starchild. You can see because the Starchild is so much denser, it makes more powder. That's basically it. So, if you take a piece of this and instead of cutting it, once you've cut it, if you take pliers and you snap off a corner, which you can do, snap it off, and now you get a nice clean edge. And if you're very careful, you can take lapidary tools like you use to polish jewelry, and you can polish that bone and you'll get a nice clear look at what's in the cancellous holes. Next slide. And what you see with human bone, this is the top, it's so big relative to the Starchild. This is the top, this is the inside. This is the same area, the same place, same piece. And it's pretty, it's like alabaster. It looks like a kind of a jewel of its own. This is human bone, normal human bone, after extended periods of death. Because what happens is in the natural course of events of life and death on this planet, Mother Nature cleans her plate, dust to dust. There are things that will eat every single aspect of us up till there will be nothing left. Nothing. There's a place in the States called the Body Farm in Tennessee where they've done extensive research on what happens to bodies. People donate their bodies and they put them out in the woods and they film what happens. And you can just see it's remarkable. Within a matter of a few weeks, you got a body laying there and you've got just a kind of a brown puddle of nothing. It's really incredible. Bones and everything. Get down to the bones, there's mold and fungi and bacteria that are going to eat that. Mother Nature cleans her plate. If she didn't, we'd be stepping out of here and we'd be looking at 1,000-foot high piles of bones. You have to understand that. Well, one of the things it does, there is a bacteria that we all carry that will eat out every single skerrick of bone marrow from our bones. Now, this is how bone works. It's like a sandwich. You got a piece of bread, and in the middle you have the cancellous holes where the marrow moves and is made and all that. Bone marrow, right? It's blood, in effect. A different kind of blood, bone marrow. So, what they do is they eat every bit of it out so much so that in the end, you could eat out of that. It's that clean. Nothing left in anybody ever. Next slide. Except for the Starchild, in which case you have this reddish residue all over the place. Next slide. We'll see a backlit piece. Look at it. No idea what it is. But it's not bone marrow. Because if it was bone marrow and it oxygenated, it would be black. You know how blood does? Blood falls, it oxygenates, it turns black. Whatever this is, it's not from Earth. It's not from Earth because Earth provides bacteria to eat every bit of it. We don't know what it is. Now people say to me, well, why don't you find out what it is? Why don't you find out what the fibers are? Why don't you find out what this is? It's very microscopic in size, all of it, number one. Number two, never been seen on Earth, so there are no protocols for recovery. Nobody's ever thought about trying to recover it before. But let's just say you could figure out how to recover it. How would you maintain the integrity of the samples that you recover? How would you get them and keep them pristine so that you could then test them? And then, because you're going to have to destroy them to do the tests, of all the thousands of tests you could run, what are you going to choose to run to destroy them? What are you going to do? You see the problems really compound when you look into doing it. And more important than all of it is, it costs an awful lot of money and we just don't have it. If there's one thing our field is famous for, it's no money. We can't access the money. The PhDs keep it all to themselves to do their stupid, flea research or what, I don't know if you ever read the crazy things they spend their money on, but nonetheless, we don't get it. They make sure we don't get it. And we have to scrounge around the way we do, and that's one of the ways they keep us down and out of the way of really bothering them. We'd love to know, and one day we will know. When it's appropriate and okay for science to deal with the star child, all of these things will be dealt with. Just like Otzi the Iceman, they've spent, what, $50 million on him over the years to find out everything they could possibly find out about him. The star child will be like that one day. Price will be no object. But not now, not until I ram it down their throats and they're forced to deal with it. Okay, next slide. All right, so this is basically just saying what I've been telling you, that the physical stuff just doesn't matter. But guess what? Recently our geneticists recovered and sequenced several choice bits of clear-cut DNA evidence, and I'll share that with you now. This is quite a ride, and it is. Now we're going to move into the DNA. Next slide.
Did you get that from the teeth?
The DNA? No. Bone. We can't. We're saving the teeth for the one that goes to film, the final. So we just get it from the bone. But the star child's DNA is good. Okay, everything about the star child skull case comes down to DNA. Now, this is where you have to start learning some things. So I'm going to kind of go slow, and I'm not going to leave anybody behind. Don't worry. All of the cells in our body, trillions of cells, not necessarily all of them shaped like a basketball, but whatever their shape is, they have a nucleus, and outside the nucleus, floating in the cytoplasm, are what are called mitochondria. There's only one nucleus per cell, and there are hundreds to thousands of mitochondria floating in here, and the mitochondria are little power plants. Inside the nucleus is all of the chromosomes, and that comes from our parents, 23 from, in our case, 23 from mom, 23 from dad, that make up 3 billion base pairs, subunits, bases, nucleotides. We'll talk about all that in a minute. But so you have the chromosomes in here, 46 for humans, and they're made up of genes, about between 30,000 and 20,000 and 30,000. 30,000 max, 20,000 the lowest amount, somewhere in there. They're still figuring out what it is. But 30,000 genes make up all these chromosomes, and each of the genes is made up of the spiral, the coiled double helix of DNA, and the DNA is made up of the key elements, or the nucleotides, these bases stuck together to make the steps of the ladder, and they produce the proteins. And we are our proteins. We make proteins. That's our gasoline. That's what we live on and move on. And also, there are power plants in the mitochondria, and they're producing power for us as well. That's what our cells do. They make the energy that lets us live. Next slide. We're going to see it in a different way. 46 chromosomes are composed of 30,000 genes, composed of 3 billion nucleotide bases arrayed in pairs. Again, that's in the nucleus, the 3 billion. And here's how it works. These things here, you have 4 bases: thymine, adenine, guanine, and cytosine. But these 4 always go together in pairs, i.e. base pairs. Thymine is always with adenine, adenine always with thymine, guanine is always with cytosine, cytosine always with guanine. So, you know how computers work with just 0 and 1? Does everybody know that computers work with only 2 basic symbols, 0 and 1? And from that infinite level of simplicity comes the incredible complexity of what computers can do, animations and all that stuff. It's all massive strings of 0 and 1. 0 and 1 is arranged in wild, weird combinations, but it boils down to 0 and 1. Same thing with us. 0 and 1. 0 and 1. DNA is the math of biology because of this. It all boils down to these 2 things. And you have that infinite simplicity blown out into the infinite complexity of our bodies, and not just our bodies, but the bodies of every living thing on earth and every living thing out there. It's incredible. It's incredible to know this. So you have that, and these make up the ladder steps here, and this, imagine, is the 20 or 30,000, the chromosomes, of course, the 46 chromosomes, 30,000 genes wadded up inside the nucleus like wads of rubber bands, just wadded up. But they're in there, and they're constantly churning, and they're constantly making themselves over, splitting, making new cells. It's mind-boggling when you really get to understand how our bodies work. It really is. Next slide. Okay, so here's how it works, except the red blood cells. All of the cells have nuclear DNA and mitochondrial DNA. And again, in the nucleus, where there's 3 billion base pairs, you have the chromosomes, 23 from mom, 23 from dad. But in the mitochondrion, which are floating out here in the hundreds to thousands, it all comes from mom. Now, how does that work? Because the egg, you've all seen pictures of the egg, how big it is relative to the sperm when it connects. The sperm is 23 chromosomes with a tail stuck on it. The egg is 23 chromosomes with all the mitochondria in there as well. So with the new person, it's got the mitochondria of the female. So all of us in this room, all of us, we have the mitochondria that our mothers have, that our grandmothers had, that our great-grandmothers had, right on back as far as you can go, with very little change over time. Mitochondria doesn't change very much over time. Now, there's an important thing you have to understand for the rest of this to make sense. In the 3 billion base pairs here in the nucleus, 3 billion, 95 to 97% of it doesn't work in a way that we understand. It doesn't make proteins. It is called junk DNA, non-coding DNA, non-functioning DNA. They're sorry they called it junk, like we're sorry we called it the star child, and they're trying to get away from junk, but still everybody knows it as junk DNA. 95 to 97% of this. So that all it's really making us operate is about 90 million base pairs. It's still a lot of room, but 90 million as opposed to 3 billion. Now with the mitochondrial DNA, it's exactly different story. There are only 16,569 base pairs. It's a very precise number. You can have 70, you can have 68, but nearly everyone who's everyone who's normal has this, and you don't want to be off normal. And it's a very precise number, 16,569. Now, with mitochondrial DNA, the same story is reversed. 95 to 97% of this works, and only a very small portion, 5 to 3% of it, does not work. And so, if you're going to have a mutation or a difference or a variation in here, and you're not in that 3%, you're going down if you're an embryo. You're not going to make it out. You're not going to be born. You've got to be normal in here, or if you have a variation, it's got to fall in that very narrow window to leave you alive and to let you push that into the gene pool of your haplogroup, whatever your haplogroup is going to turn out to be, because you now have a variation that's going to make a new haplogroup, or part of a new haplogroup. Stuff we'll talk about in a minute. So you can hit in here with mutations in that 95 to 97%, and it's not that bad a problem. Only when you get into the small portion that works, if you have a mutation there, do you begin to have problems. And guess how many we have? Over 4,000 genetic disorders in our gene pool, making us the most plagued with genetic disorder species there is. And that's for another lecture, but it's basically because we're genetically engineered. We're not a natural species on this planet. Anyway, next slide. That's my other work in human origins and hominoids and all that. This is an early gel sheet where the early part of DNA was where you were recovering with primers, 20 to 30 base pairs long, very tiny little strings, and you had to know what it was. Just understand that in the early days of DNA, it was the Stone Age compared to where it is now. Next slide. First DNA test done on the star child in late 1999 was done because there were 6 labs in the world that could handle what's called ancient DNA. Ancient DNA is dead longer than 50 years. Star child was dead 900 years, so it was definitely ancient DNA. But to do ancient DNA, to handle it properly, because it's so fragmented, it's like, you remember the wad of rubber bands? When it's been dead over 50 years, those rubber bands have been put through a wood chipper, and you've got pieces the size of glitter scattered on the floor. And that's what you've now got to pick up and recover and figure out. You can do it, machines can do it, computers can do it, but that's what you were up against back then, and this lab couldn't do it. It was a forensic lab. Those 6 labs could, because here's why. If a flake of skin off of one of the experimenters, a flake of skin hit the solution, ruined. If a droplet of their breath hit the solution, ruined. Right? It was like you had to work in a clean room. That's why there are only 6 labs that could do it. I wanted some kind of result, so I gave it over to these people. And they started out, I think, with the best of intentions. But in the end, other people on the campus found out what they were doing, and they just put pressure on them and said, get that thing out of here. Just get it out of here. And so, next slide. The report I got from them finally after 2 contaminations was a very weak result, 200 picograms, when 1,000 was normally what you would need. And they said the result is XY chromosomes, which tell us the star child was a male and it's human. They were basically saying, go home and forget about this. This is dangerous. We can't fool with it. You shouldn't fool with it. Forget about it. And I said, you know, this can't be right. And they said, it is right, and we're going to put it out. Now, they again had already contaminated 2. This is another contamination. I don't even know if they even did it. I think they just wrote this because this was what the mainstream scientific community wanted to hear. And right now, to this day, again on Wikipedia, you will read them referring to this test right here from Bold in '99 saying that the child was male and that it was a human. You can read that as if it happened yesterday. As if it happened yesterday in criticism of the star child. And so we were just absolutely floored. When this happened and the word went out, everybody, everywhere heard that the star child was a human, and that was it. Next slide. And so for the next 3 years, it was dead. I couldn't get anybody to listen to it.
Lloyd,
Yes.
who funded that lab who gave that information? Who were the main funders?
The owners of the skull, Ray and Melanie.
No.
The first lab was funded by Ray and Melanie. Yes. I'm getting